In this lecture, I'm going to talk about the basics of the synapse. As you probably already know, synapse is just going to be the connection between one neuron and another. Typically, it's going to happen at the terminal bouton. And a dendritic spine. So in this case, here we have the terminal. Here we have the dendritic spine. And you can see there's many synapses, many connections on a typical neuron. Each axon branches and forms more than one synapse also. In the central nervous system, there's about 10,000 synapses per cell. Okay. Although mostly it's between a bouton and a dendritic spine, we can have them pretty much anywhere. For the purposes of this class, when we think synapse, though, this is the connection I want you to think about for now. When the exceptions arise, I'll let you know. Well, the way we know that there's synapses is because the amount of time it takes for a signal to get from the brain all the way to the muscle is longer than if it was a direct connection. So the fact that it's slower tells us something else has to be going on. Around the turn of the century, turn of the 20th century, Kalat and Camillo Golgi, I'd mentioned them a little bit, talked about the structure. They used Golgi stain to catalog neurons. And that is part of how we knew they were discrete. And that this connection, there was a functional connection. Now, when we have a connection, it can be two different types. It could be a chemical or it could be an electrical connection. We weren't really too sure of the nature of this connection. In a minute, I will show you an experiment that helps settle that. In the body, this is a more accurate look as to what happens. We've got the cell body, we've got the axon, and then we have a bunch of terminals making synapses onto primarily dendrites, but also some other places. When I draw it, I'm going to use a simplified representation. I'm just going to draw a circle, a line, and that little thing. Okay, so this, I'm not going to draw all the synapses. I'm not going to draw all the dendrites. I will draw the axon. And here, I'm just going to draw and make it look like there's only one connection. Okay, but as you know, if we were to look under a microscope, there's many more connections. Now, there can be two types of synapses. One is the so-called chemical synapse. The other is the electrical synapse. This is also called a gap junction. In the central nervous system, the vast majority of synapses are chemical synapses. And when we are talking about psychology, the synapses we are primarily interested in are chemical synapses. Okay, so I'm going to spend almost all our time focusing on chemical synapses. I do want you to be aware that electrical synapses do exist. Okay, I haven't told you what those are yet, but I'll explain that at the very end here. All right. So how do we know that the connection is chemical? Well, there was an experiment. The vagus nerve uses acetylcholine. At the time, they didn't know it, but we know this now. With this particular experiment, if you stimulate the vagus nerve, it's going to send signals, and that's going to slow down the heart rate. Okay, so if I take electrodes, stimulate the vagus nerve, my heart is going to slow. We didn't know the nature of the connection between the vagus nerve and the heart muscle. Okay, this space, this connection, we didn't know if it was chemical or electrical or what. Well, it turns out if I take the fluid after I stimulate the vagus nerve, I just take the fluid, there's no stimulation, and I transfer that to another heart, I can slow the heart. Okay, so what that tells you is that when I stimulate the vagus nerve, it's going to release into this fluid some sort of signal that causes the heart to slow down. I don't need to stimulate the vagus. I just need to take that fluid and expose another heart to it. When I do that, then the heart will slow down. Okay, so that tells me it's something in the fluid. That means it's going to be a chemical signal. Okay, so Golgi and Cahal told us that there were connections between neurons. This particular experiment taught us that at least some of those connections relied on a chemical signal. Now, in this intro, I'm going to kind of give you an overview. I'm going to tell you about the whole chemical synapse, start to finish, what happens, and then I'm going to go into more detail about the events in subsequent videos. So, remember, we will have our terminal bouton, making a connection with a dendritic spine. Okay? So somehow we have to get a chemical signal from this cell to this cell. And remember, we've got the action potential. We talked about that. We talked about propagation. Right? When that electrical signal hits this terminal, now somehow that has got to be changed into a chemical signal. And that chemical signal is going to have to come across this space and have an influence on the second cell. Okay. The cell before the connection is the presynaptic neuron, presynaptic cell. The one after is the postsynaptic cell. Okay, so somehow I have to transfer information from the presynaptic cell to the postsynaptic cell. All right, so we'll begin with the action potential reaching the terminal. At the terminal, there's special voltage-gated calcium channels. In response to this depolarization, calcium is going to come rushing into the cell. Okay? So, the action potential depolarizes the membrane. That causes those voltage-gated calcium channels to open. Calcium flows into the terminal. The reason for that? There's a higher concentration of calcium in the extracellular fluid. Okay, so the calcium is going to flow down its concentration gradient and come into the terminal. When the calcium comes into the terminal, it's going to fuse with synaptic vesicles. It's going to interact with synaptic vesicles. Those vesicles then will fuse with the cell membrane. So a synaptic vesicle is a membrane sphere. Inside it, I'm going to have chemical called neurotransmitter. Normally, that's going to be floating around in the presynaptic terminal. When calcium comes in, it's going to interact with this, and it's going to cause this to fuse. It's going to move down to the membrane and then fuse. When it fuses with that membrane, that's going to cause the contents to spill out into the space. Okay? So calcium causes the synaptic vesicles to fuse with the membrane. That's going to cause the contents to come out. That's then going to diffuse across this space, which is called the synaptic cleft, and interact with proteins on the postsynaptic cell. these proteins receive the chemical signal. They're called receptors. Okay? So the chemical that binds to them is called ligand. Okay, so in this case, whatever neurotransmitter, whatever chemical this is, it's going to diffuse across the cleft, and it's going to interact with this receptor. As long as there's neurotransmitter there, chemical there, interacting with the receptor, I'll have an effect. Okay, so whenever ligand binds to the postsynaptic receptor, it's activated, and there will be a postsynaptic effect. Now, we don't want this to go on forever. So over time, we have to terminate that signal. There's a couple ways we can do it. One is we can destroy our chemical signal. And that's called metabolism. So an enzyme destroys the signal. So it destroys the neurotransmitter. Okay, this is a mechanism by which acetylcholine, which we'll talk more about, acetylcholine signal is terminated. We can also do reuptake, where special proteins in the presynaptic terminal grab the chemical and bring it back up. Okay, so this is kind of a recycling. Things like dopamine and serotonin use reuptake. Okay, so those are the basic steps. Action potential hits the terminal. Voltage gated calcium channels open. Calcium rushes into the terminal. That calcium is going to interact with synaptic vesicles. It's going to cause a cascade of biochemical reactions, causing those vesicles to fuse with the membrane. When the vesicles fuse with the membrane, they release their contents into the synaptic cleft. The neurotransmitter then will diffuse down its concentration gradient, and it's going to interact with postsynaptic receptors. When it interacts with postsynaptic receptors, it will activate those receptors and will have a postsynaptic effect. We don't want that signal to go on forever, so somehow we have to terminate it. We can do that by breaking apart the neurotransmitter, or we can grab it and recycle it, get it away from the receptor. Okay, that's a basic summary. Now, there's a little bit here, and then in the subsequent videos, I'm going to go into much more detail on these steps. The synapse is a very specialized structure. So if you look at this, this is an electron photomicrograph of the brain. Here is a terminal. I know this is a terminal because I can see synaptic vesicles. Okay, so these are synaptic vesicles. I can also see here in the postsynaptic cell something called the postsynaptic density. Okay, you see this dark stuff? You see that on the postsynaptic side of synapses. If you look here, I've got the membrane from the presynaptic cell. Here I've got the membrane for the postsynaptic cell. And you can see that little space. That little space is a synaptic cleft. Okay. Here I've got the vesicles containing neurotransmitter. You can't see the neurotransmitter. When calcium comes in, these will fuse, neurotransmitter will be released, and it's going to interact with the postsynaptic receptors here in this density. Okay, so the reason this is dense is because there's a concentration of receptors, which you can't see. But there's a concentration of receptors and other proteins there holding those in place. Okay? So, sort of taking a look at chemical communication and even a larger picture. We're going to look at how we get neurotransmitter. The first thing is we have to synthesize and make neurotransmitters. Okay, so vesicles are going to come down from the cell body. Some chemical signals are made in the cell body, others are synthesized here in the terminals. Okay? But smaller neurotransmitters like acetylcholine are going to be made here in the terminal. Once we make it, we're going to store it in these vesicles. Action potential is going to cause calcium to enter. That's going to result in the release of neurotransmitter. Neurotransmitter is going to diffuse across the synaptic cleft and bind to receptors. As long as we're bound to these receptors, we'll have an effect. Okay? Now, these don't stay bound forever. It's not a covalent bond. Think of it as a handshake. This receptor, this neurotransmitter is going to come. It'll shake hands with this receptor. While it's shaking hands, the receptor's activated, and we have an effect. Over time, it's going to let go, and my neurotransmitter then is going to float around. It's going to keep floating around until it comes into contact with another receptor. There it's going to shake hands with that receptor and activate it. And it's going to keep doing that until something happens to it. Okay, so in this step, my neurotransmitter has become separated. Now, once it's separated, a couple things that can happen. In this particular case, I've got a protein that's going to grab it and recycle it. It's going to bring it back into the terminal. Now, when my chemical is brought back into the terminal, it can no longer interact with the receptors. That's how we stop the effect of the neurotransmitter. Now, there's a couple extra steps here that I didn't talk about that also can occur. Sometimes this postsynaptic cell, if it's getting stimulated a lot, may release a signal that's going to come back, and it's going to slow down. It's going to inhibit this cell. Okay, so when we inhibit the release of neurotransmitter, we're going to exert a so-called negative feedback. Okay, so there's a couple things that can happen. One is neurotransmitter from this cell can be released. That's going to go backward. That's why it's called retrograde. Retrograde signal will tell this to kind of calm it down, stop releasing so much neurotransmitter. We'll learn later there can also be receptors here that will respond. So when this neurotransmitter is released, it'll actually bind to receptors on the presynaptic terminal. These so-called autoreceptors will then inhibit the release of neurotransmitter. Okay. Now, one of the things to notice too, we got a glial cell here. Glial cells are actually quite active. They can take up neurotransmitter, they can release neurotransmitter. There's a lot they can do. And we'll talk about specifically what they do down the road a little bit. So now again, with the chemical synapse, action potential reaches a terminal. Voltage-gated calcium channels open, calcium comes into the cell. Calcium causes those vesicles to fuse with the membrane and release neurotransmitter. The content of the vesicle then diffuses to the postsynaptic receptor where it's going to have an effect. We want to terminate the signal at some point, so my chemical is either broken down by metabolism or taken back up into the presynaptic terminal. Now, these steps are important for you to know because drugs affect every single one of those steps. And in this course, you're going to need to be able to predict the effect of a drug if I tell you what it does. Okay. Here we look at a dopamine synapse. Okay. Dopamine, we'll talk more detail, is made from tyrosine. Tyrosine is going to get converted into a chemical called dopa, and then dopa is going to get converted into something called dopamine. That dopamine is going to get packaged into vesicles. When we have a depolarization, calcium comes in, vesicles fuse, we'll get the release. Okay, that release then is going to allow the dopamine to interact with receptors on the postsynaptic cell. Dopamine would normally get taken back up and broken down where so it can no longer have an effect. Okay, so there's lots of different drugs at different things. So if we increase the amount of dopa, I give a person dopa, which is a drug, that will lead to more dopamine. If I have more dopamine, I'm going to be able to make more. I'll have more to release. When I get a action potential, more neurotransmitter may be released. Some drugs are going to block the receptors. So if we block the receptors, instead of having a nice spot where dopamine can bind, right? If I have a drug that blocks that, now dopamine can't bind and can't do anything. Okay? Other drugs block the reuptake. So if I have a lot of neurotransmitter floating around, and normally we halt the signal by bringing it back up into the presynaptic terminal, right? That would change if we have a drug like cocaine. Cocaine blocks that reuptake. So now, instead of terminating the signal, this step doesn't happen anymore. If that step doesn't happen, I'll have more neurotransmitter. If I have more neurotransmitter in the cleft, I'll have a bigger effect at the postsynaptic cell. Okay, so I just want you to realize that there's many, many different drugs. All of them will affect different steps in this chemical synapse. So as we go through, you'll learn specific drugs, you'll learn which steps. But the first thing is to go back and memorize. Make sure you learn the steps of neurotransmission at the chemical synapse. Thank you.